introduction. Good morning, everyone, and good afternoon and good evening, those who are listening online. It's a great honor to be here. As Victor said, we go long, almost 10, 15 years together. It's good to come back to Barcelona. I was just telling some colleagues, I came to Barcelona in 2007, of course, with my wife, and this is my second time. So it's a great country, great city. Love to come back again, and thank you for having Steve, me, and of course, Michael. So, very short answer, if you want to really hear how physical form affect pharmaceutical development. 30-second answer, but I'm going to talk for half an hour, right? The 30-second answer is different physical form have different solubility, dissolution that affects absorption, bioavailability, and dose required in human and preclinical species. So it is very important that the physical form has to be stable and it should not change. That's the final answer, right? So I'm done actually, you can save 30 minutes, just kidding. So no, this is really, really important topic and very near and dear to my heart. So what I'm going to talk today, I'll just roughly give an overview about the drug development process. Majority of you know because you're all from industry. And what is BCS classification? Because that connects with the physical form. Everything we are talking, they're all connecting with the physical form. And then what exactly physical form means, of course, you will hear from other speaker as well as Steve, how that crystallization and whatnot is important to obtain a stable physical form. But I will talk about basic fundamental, how that form plays a critical role. And of course, some technologies to improve solubility and dissolution of those physical form and formulation. And I'll just summarize my talk. So as you see, early on in drug discovery, there are, it's like more than 10,000 candidate goes through molecular modeling. Now you can say machine learning. Now you can say AI. Whatever you use. So more than 5,000 to 10,000 molecules we put together. It takes almost 3 to 6 years, and from there you can imagine 250 enters into preclinical phase where pharmacology and tox studies are done. PK studies many molecule, but pharmacology and tox around 250, and then you can imagine it enters around 5 molecule in the clinic. Just a rough idea. It could be little more plus minus, but get an idea. So where we start with 5,000 to 10,000, enter in the clinical around 5. So very high attrition rate, because we have to select the best candidate. And then finally, after 7 or 8 years or sometime even 12 years, one molecule gets approved. If you are lucky, right? That's very important. I don't know how many of you have served industry for, well, with Novartis, we have a big pipeline, and I'm sure with previous experience from Michael, GSK, we have a big pipeline. So I got to see almost, I would say, I'm with Novartis now almost 18 years, and in 18 years, almost 22 molecule I had a chance to see going from preclinical to commercial, which is a blessing, true blessing. But I do stop at a certain stage. I don't take all the way up to, late phase, because my team is focusing on early part. So you see how but then while the number of molecules going from 10,000 to 1, how much it cost? That is also a good idea to know. Rough number, right? And you can see the answer is you can see answer right here, depending on the scenario, which is very niche, rare disease, right? Then you could wrap up all the study in $1.5 billion and 6 to 7 years. But if you're going for cardiovascular or cardiometabolic disease such as hypertension, diabetes, there you need 10,000 patients. You need 2 to 3 years of study. It's a huge study, and it's longer time also. And there we may end up being almost 4.5 billion. And you know that especially when patient receives the cost, you'll say, oh man, this medicine is so expensive. why it is so expensive? And one of the reason, first of all, one medication almost takes 4.5 billion, but 10 medications fail before it gets approved. So imagine, we already lost 40 billion. So somewhere, and don't want to say that, somewhere they have to, they have to recover a cost, right? They means all the pharma, because you are spent, So long story short, that's why medicines are very expensive, But that's not important. The most important is saving and extending patients' lives, as you all know. That is very important. Money will come, money will go. We have this one new medication, gene therapy. A lot of gene therapy have come now. If you follow the gene therapy, there are like 7 products now. But 3-4 years ago, we launched camera, which was almost $2 million, and everybody was saying, 2 million? That's too expensive. But it's just one injection, and that's it. The kid below 2 years, you inject that, and then all the muscular dystrophy, because that disease is after, if you do not inject before 2 years, what happens is by 12 years or 14 years, your muscle won't work, and the kid has to, give his life, his or her life. So below 2, if you inject, then you are good for rest of your life. So in reality, that's a life-saving drug. It's not the cost, it's like, saving a life. So just to put into perspective how much it takes to discover and develop, and ultimately how much patients are benefited. But however, what happens because of all this, it is very important that the message keeps coming. Can you shorten the time and reduce the cost? That is always ultimate goal for every industry. Reduce the time, cut the cost, get into the faster. Into market. Now, with that, how many of you are aware of BCS classification? Couple, a few, almost, yeah. So I don't go into more detail. This is very important for any drug developer, for any formulator, for any PK scientist, DMPK scientist, anyone who is in industry must or should know about BCS classification. And that actually ultimately we classify this at phase 2 or post phase 2, because that time you have dose is defined or finalized for phase 3 and commercial. However, you have a good idea that, if you see most of the drugs in the market, of course, doses are like 5 milligram, 10 milligram, but very common dosage, if you see, between 100 to 400 milligram. 80 to 90% of the medication, especially in oncology and other area. And majority of the industry is working in oncology. If you look, in Boston, we have 600 biotech and 350 are working in oncology. So an average dose you can see from, 200 to 400 or 100 to 400 milligram. So early on, when you do pre-screening, you should have an idea that could be the dose, and then accordingly you decide your roughly you can say pre-BCS or DCS classification. So that way you know what complication you are going to have while developing. I wouldn't say complication, but what kind of path forward you could take for this molecule to develop, what kind of physical form is required, and that you will see. And that's what I thought, good idea to give a BCS classification, So in this you will see class 4, right? You saw, there are class 1, class 2, class 3, class 4. But you can see the class 1, you are very lucky. This is an old one, almost like 10 years. Right now, if you ask anyone, have you ever seen class 1? They will say, not in last 10 years. Have you seen class 2? All the time, right? So mostly you will see molecule in class 2. That is the, usually class 3, solubility is very high. Perhaps you can see class 1 and class 3, and you don't see very high solubility. So anyway, but if you see, then it just differentiation is permeability. And for class 2 and class 4, of course, you can see the solubility is affecting. For class 4, permeability is also affecting. Now, a quick question here. If you see, like, if early on you're working in research and someone comes with class 4 molecule and will say, well, I have low solubility as well low permeability. What do? I spent 3 years. Chemist, maybe dear chemist, if somebody's here. And they'll say, well, what is the efficacious dose? 300 milligram per kilogram. Do you have the molecule? That's a very high dose. Can you work on potent molecule? That would be your feedback, So that's why the class 4 molecules are not many in the market. And we should not drop. If someone comes with like 5 milligram dose or 10 milligram dose with class 4, so what? It's a molecule. It's even 5% bioavailability is giving you what you need. Proceed. So that's really intellectual interactions you need to have between chemist, biologist, formulator. That is very important. And of course, physical form, formulator, they are all come together, biopharmaceutics. So do not throw the compound, do not drop the compound, have early on interactions, involve a solid state team early on, what we could do to make this happen, even if it is class 4. That's about the classification, BCS. Now we'll switch the gear. Now you have an idea, drug development, you have an idea about the BCS classification. And in solid state, this is very basic slide. The physical form, organic solids exist in crystalline and non-crystalline. And you can see in crystalline form, single nice crystalline material, multi-component, we have salt, you have co-crystal, and then there you could have hydrate, solvate, and others. And there are different polymorph possible, because depending on, sometime if you must see or have seen one molecule, and you will show every time X-ray is different, DSC is different, what is happening? So there, most of the time, they are either solvate or hydrate. And there could be different form, they considered as polymorph. But real polymorph could be 4, 5, sometimes could be 30 also. So it is very important to understand, if you are having a development candidate, you need to understand that risk and involve a solid state colleagues early on and say, hey, what is the risk? This is a big risk for me taking into development, and these guys will advise you really well that it's not a big risk. We can manage moving forward. We can identify the stable form or stable salt or co-crystal, and we can take it further into the development. Is it a go, no-go? Perhaps majority of the time it's not a no-go, but then keeping one physical form stable all the time from preclinical to clinical is the key. If the form keeps changing, FDA or authority will say, well, you are not keeping a track of your form, and that would have impact, as I said in 30 seconds. That would have impact, and then how do you control your drug product? And they will say, wish I could have invested early, or more time understanding. So that is the basic actually. And this is a very simple. If you go to London, this is London. It's all short range order, amorphous form. Can vaporize anytime. No, not London city, but the amorphous form, unstable. And this is New York City. Very well planned, street, avenue. And if you want to define, that's kind of crystalline form. Define order. Just gentlemen, layman, kind of to understand what is amorphous, what is crystalline. And you will say, well, how do I differentiate those forms? X-ray is the ultimate, X-ray will tell you exactly, but this is a DSC, and there you will see free form has a melting point of 201, and one of the salt has 253, another salt has 283, which means if you have one compound free form and we have different salt form or different polymorphs usually you will not see there's a big differences. Polymorphs are lingering around couple of degree here, couple of degree there. It's not much difference on polymorphs unless there is, a huge difference. But salt form, huge difference in melting point and of course XRPD. But that's not only the thing, but it's one of the technique where someone says, how do I differentiate, that this is one of the technique to find out. And then solid state colleagues have this all detailed information on X-ray, DSC, TGA, so they will take care of, you just understand the impact. Now, when these different forms we kind of select or identify, how do you manage their solubility and dissolution? Amorphous form, if we have, or solvate form, you will always get higher solubility and dissolution. And you will say, oh, I'm all set, I have good solubility and dissolution. The minute it comes to Solitek, And then they identify and they'll say, oh, we found a nice stable form, solubility is 5 microgram. Are you kidding me? I was getting more than 200 microgram or almost 1 milligram, and now what's happening here? Well, now we identified the right form, 5 microgram, and then what do I do? Then these colleagues will say, don't worry, we got the solution. And these are the different solutions, you know. So these are the different technologies we could use when we identified the stable form. We could use this technology without identifying the stable form, but that will not tell you anything. So that's why I think early on, before you enter, the question comes, when do I lock the physical form? When do I select the final physical form? Now, I'm really big fan of like identifying and locking the physical form as early as possible, so that there are no bridging PK, there is no repetition of talks, there is no repetition of formulation. Why you have to repeat everything? I mean, the patients are waiting for medicine, and here we are just repeating. I mean, anyway, you have to spend money. You spend later or you spend now, and get things done, science right away. That's how I am. In my team, we try to finalize physical form as early as possible, even from 2-week mini talks. That's it. What you use for 2-week mini talks, 4 weeks GLP, and clinical. Because it's the right science, doing the right thing at the right time for the right science. It doesn't matter. Someone says, well, I don't know whether a molecule will survive or not. I can do that later. You can do that anywhere you want, but that's not the point. If you really want the molecule to go faster into the clinic, to the patient, do right science at the right time. And these are the different technologies I won't mention to take care of a physical form which has a 5 to 10 microgram solubility, right? Class 2, typical, going back to our BCS classification. This is again another most important. All the formulators, solid state, that kinetic solubility plays a key role. Every molecule should have a good dissolution rate, high solubility. And every molecule brings that, more or less. However, it crashes down right away. I get good solubility, but it crashes down. And then, what do I do? What you need is, you need a solubility, and then you need a parachute. You need solubility dissolution, then you need a parachute. At least for 4 or 5 hours from the dosage form. But for preclinical studies, this doesn't help. You need in a solution for a couple of hours or even couple of days. And that's why selecting the physical, sometimes you might get it right away. For human studies, you're fine, but for preclinical, which is my team job, it's more tough. They will say, well, what is this? I'm not making formulation every day. You give me something stable for 7 days, 2 weeks. So that's even tougher. And higher doses, even tougher. We need to dose 300 milligram per kilogram. You must have seen it. We need to dose 1 gram per kilogram. And in clinic, your starting dose is 5 milligram. In preclinical, 1 gram. I say, what? In clinic, you need 5 milligram. Because in preclinical species, you want to understand the safety margin, therapeutic window as much as possible. And that's why the doses are very high in preclinical. And these are the techniques. You know, in nutshell, these are the techniques will help you to get the parachute. I am a big fan of salt. Any techniques here, they are all good, by the way. But someone asked me in last 25 years, Sudhakar, if I could make a salt and maintain that parachute by some surfactant or excipient, easy life. Why you want to go to all this painful, time-consuming, money-consuming, and then end of the day you will say, well, that's not stable either. So the first thing, and if you see market, and I might have slide, there are like out of 2,000, there are 1,500 salts in the market. Hello? Right? Why? Because that's the easiest path forward. If molecule has a potential to ionize, we should take benefit of it. Or you should design a molecule. Who are the formulator solid state here? If you interact with chemists, you will say, do you mind adding ionizable group? Well, I can add, but it will affect my potency, it will affect my efficacy, it will affect my toxicity. Well, have you ever tried? Can you just put it and see it? Yeah, let me try. And if it is not affecting, win-win situation. So that's one of my role actually, my team's role. We work with chemists and design a molecule accordingly so that we find a simple, easy path forward. Or co-crystal, easy. make it life easy, you know. So that's how one of the way of interacting with your biologist, chemist, to design a molecule depending on the dosage form. And these are the different scenarios. So very briefly, I can tell you that if you are working, so there are a couple of scenarios, and you might say what is high and low. So you will see the high means bioavailability 80%, low means this, and medium means this, you know. So if you dose solution and suspension, and you get couple of scenarios. So this scenario, solution and suspension at pharmacological dose, is class 1 and 3. How many molecules do you have? Barely. So win-win situation. You go ahead with this route, like simple, easy. The contents are missing there, but it's easy to develop, no problem. Class 1, class 3. Here, class 2 and 3, majority scenario we are here, which is class 2. It's very important that what is the maximum absorbable dose and what, is there a food effect. So if you understand, if you are here and understand this, then you have to decide which technology you can use. And if it is class 4, as I said, you want to really understand this poor exposure is because of first pass metabolism, so you can understand the portal vein study, or permeability. So this is a very basic slide actually, and it's super powerful for any formulator, or you know, this you will not get anywhere. This is our experience from last 20 years, tells you for formulator, for any, once you classify this, which kind of technique I could use to move further, systematically done. That's why at Novartis, when we have been working in last 20 years, we recommended for 600 candidate, we recommended certain formulation principle based on the preclinical data for clinical. Once we entered in the clinic, 3 times we had to change the formulation. Out of 280 compound we took into, only 3 times. Not bad, right? I mean, means we know what we are doing actually. You know? So, in nutshell, because it's a scientific driven decisions. It's not like magic or something. It's a science driven decisions. What is solubility? What is dissolution? What it translates into the preclinical? And then you do the modeling, GastroPlus modeling, predict the dose, and you recommend formulation for clinic. And that's how it is, you know. So this is very powerful slide, by the way. And this one is like how do you calculate the maximum absorbable dose? Now I gave you very high level that 200 to 400 milligram dose is an ideal, but this gives you an idea that when you have, you know, different species, and you calculate the maximum absorbable dose, and you will get the, you know, MAD. So very high level you can see depending on the solubility of your molecule. This is ready-made, you know, and you saw the formula. So this is the ready-made. If you have this solubility, you will see these are the doses perhaps with the 90% fraction absorb. So you can say that, oh, if you give me 1 milligram solubility, I can dose up to 4 gram, almost 3.5 gram. Or if you give me really, ideally, if you just need, like, you know, for preclinical, clinical between 0.1 to 0.5 milligram, just an example, and then you are very much covered between these doses. So, you know, just to take away. You want 0.1 milligram solubility in FASU conditions. That's the standard, gold standard, based on all this experience. Someone says, how much solubility? Chemist says, Sudhakar, how much solubility do you need? Depending on dose. We don't know the dose. Well, nobody knows dose. So ideal scenario, 0.1 milligram at physiological condition. That's the gold standard. And you will not go anywhere wrong in preclinical when you dose as suspension, and in the clinic when you make a tablet or capsule. Very simple, 100 air volt technology. Reduce a particle size, you will get the exposure. Surface area increases, dissolution increases. Very old technique, right? Salt approach, one of my favorite approach, as I said. And look at this. These are very old molecule. Codeine as it is has 8 milligram solubility. Why do I need a salt? But we never know. Because codeine syrup, most of the cough syrup have codeine, and you need high solubility there, 100, 200, 300 milligram. Look at that, phosphate. So if you see codeine syrup, codeine phosphate, this much. Although you have 8 milligram, I just said you need 0.1 milligram, you got 8 milligram. Why the hell I would make salt? You do not know in future what you need. So always plan for future and see. So that's why I'm a big fan of, kind of salt. You get an idea where I'm going, if the molecule has potential. A prodrug approach. Prodrug approach is also very helpful for solubility, but 99% of the time, prodrugs you make for improving your permeability. If someone says, my molecule has a poor permeability, can you do something? Yeah, I can add permeability enhancer, but that moves needle. If you have 10% bioavailability, goes 15. You have 2%, goes 5. If you have 20, goes 25 with 100% variability. Right? I mean, those are the permeability enhancer. So if early on, if you have a potential to influence chemists that we can make a prodrug, go for it. And prodrug helps a lot reduce permeability. Prodrug comes with liability because chemical instability and all that. But there are a lot of prodrugs in the market, so you should not worry. That would be my kind of takeaway on this slide. Another technique is solid dispersion. You might be aware of solid dispersion, highly metastable, unstable, but there are more than 20, 25 products in the market, more than 150 in the clinical trial, and solid dispersion has become a normal now. It's no big deal. We understood the mechanism really well. So do not hesitate if you have to use it, okay? And it's kind of a drug, substance, polymer. You use organic solvent. If organic solvent you don't want, you want to be green chemistry, go for hot melt extrusion. But molecules should have potential to go through that route, hot melt extrusion. You must have heard that, So on solid dispersion, there are 3,000 publications, and we can talk whole day just like how we can do it. But that's not the point is, you can go there if the solubility is poor. And this is how we do it. Because very briefly, you can have a number of polymer and combination, and in our shop, we just kind of, high level, take a couple of polymers, understand the solubility, dissolution, and see, and then pick up few and scale up. That's one of the technique, to select the right polymer. Now we did like more than 600 candidates. We know there are only three, 4 polymer works and couple of drug load. So we nailed down that. We don't need to do like 96 well and high throughput, because that's the experience we build in. Another, like this you can see the case studies. You can see it drops off in, when you change the method, but you want formulation to stay straight. And when that happens, you can see you are going to get good exposure, which might be on another slide, if I'm not wrong. Yeah. So you can see there, how that helps in getting the, exposures with the crystalline and the solid dispersions. And this is just to, as I said, there are a lot of products in the market, just to get a reference. There are a number of products. And these slides, I think Victor can make it available for you, those who are interested, so no big deal. Just don't put on the net or anywhere, you just keep it yourself. It's approved. So, see, I told you the example, and this is from Pharmaceutical. We use that Pharmaceutical very often, just to get an overview. See, this is all solid state. Our colleagues can do magic for you. I mean, certainly you can do it, this, but if this gives you a solution, you are good. Look at this. How many products in the market, right? And then counter ions. We were just talking, like, very small molecular weight, easily, if you can make it. Our stomach has hydrochloric acid. Nobody will say we have a safety concern, right? Your body has that. So our PCS colleagues will be happy to see this, those salts, counter ions. This mesylate, tosylate, one time when you consume with alcohol or use ethanol in crystallization, they may form sulfonate and they are carcinogenic. So you have to just stay away from alcohol, right? And that which is difficult, but we have during crystallization. I mean, Okay, so I think right on time. Just to summarize, as this is focused on more polymorphism, I'm just kind of focused more on, so you can see the polymorph screening is important to avoid unwanted surprises. Suddenly we are going in the clinic and I got new physical form. What do I do? Well, now you lost another year. That's not good, right? So it's very important to make that happen sooner. Salt screening, you heard me a lot. Co-crystal, improves dissolution rate. It doesn't very much, but 2 to 3 fold. And sometimes what happens, co-crystals are good for dissolution, sometimes just to improve phase chem property. If compound is just amorphous and very sticky, if you make a co-crystal, free-flowing nice powder, why not? You don't want sticky material lingering around and losing. So just make a co-crystal for better phase chem property. And you saw the several technologies used for improving the dissolution and bioavailability. And it's very important that any technology we use, the molecule has to be all this: stable, reproducible, good product profile, cost, right? And you can see importance of the physical form. And then lastly, this is of course Novartis colleagues, my advisors, and most importantly all of you coming here and supporting, of course, and learning and taking some good messages from here. So thank you so much. Really appreciate it. Thank you. Not sure we have time for questions. Okay. Oh, so sorry.
Thank you very much, Sudhakar. Thank you, Sudhakar. I'm going to open now for questions in the audience. Is anyone got any questions?
Yeah, please.
So I know it must be soul-destroying to see how long it takes for so many drug candidates to find their way to a phase 3 trial and then eventually lead itself to a drug product on the market. Soul-destroying, because you're quite right when you say it's the patient that are waiting for these drugs.
Correct.
And so many of us even today have some underlying condition where these drugs are not quite available. And then, of course, you're also reading in the market that some of these drug products take so long to develop that a course can cost $1 million for a course of drugs, which very few people can afford. So I say to you, during the pandemic, where there was an unknown virus and things could be done so much more quickly, and the solution to an unknown virus was found within a year of the virus killing so many people, why can't those changes be made in the approach on how one would develop new drug products in the way that you've described?
No, I think it's a very good point, and this question has come a lot actually. Thank you so much. Excellent point. In emergency, we came up with less than 1 year, of course, a vaccine. Why not we develop all other drugs in that short time? And it's a not straightforward answer. Even for vaccines, you see most of the influenza or viral vaccines, as soon as in eastern part we see which is the virus there, and within 9 months we can make our regular vaccines. You make it and it's available to Western countries right away within 9 months. But that's known safety, known toxicity, more or less, depending on the bugs every year is rising. But for most of our new molecule, that's not the case. What FDA wants to know, and 50 years ago, we tried to do shortcut, and this thalassemia situation and limb deformation and all that happened. So the regulatory authority, they really do not want to take a shortcut. Again, you're right. I mean, that's one answer. Safety has to be established depending on the duration of therapy. But most importantly, that if your molecule gives proof of concept, as soon as you enter in phase 1 and you have a positive POC, authority says how fast you can go to commercial. Make that happen really because it's working in patient. I do not want to see 8 years again in phase 2, phase 3. You make it happen, especially life-saving drugs, oncology, cancer. And we took those molecules in less than 5 years to commercial. So depending on the scenario and unmet need, we can go faster, but not within 1 year for small molecule, unfortunately. I hope that answers. We would love to have it, but because of the safety, you have to understand the safety, not only in preclinical, but in human also, to avoid long term. So vaccines, we took it, but everybody is worried. I don't know after 10 years what will happen. I took it. Nobody knows. But most likely, they are messenger RNA, it's in the body. It's not going to mess up. 99% we are fine, based on the science. But some people, they are against vaccine. They still worry, like, oh, good, I didn't take it. But we are in 99, so we will be all good. That's my understanding. Thank you. Any other question before I'm here, so happy to discuss and,
Any more questions? Okay, Bashir, Bashir, no? Sudhakar.
Sure, thank you.
Bashir just asked a question. I got a question for you. You showed a slide in which you compare, I think it was codeine, parent compound with two of the salts and a significant increase in solubility. But you talk about water, okay? The GI tracts go a change in pH, and obviously that's going to have an influence on ionizable molecules. What is the main drive when you are selecting a salt candidate? What actual parameters are you looking at rather than maybe solubility in water?
Very good point. I mean, water solubility is pH driven, right? I mean, if you have a salt, you dissolve in water, you will get 10 milligram or 20 milligram per ml. And if you measure pH, pH is 2. You say, pH 2, what do I do? pH 2 from stomach, I mean, nothing absorbs from stomach. So very good point, Victor. Solubility in water is good when you want to make syrups, when you want to make parental dosage forms, and you measure the pH. However, the real solubility is from pH 4.5 onwards. So pH 4.5, 5.5, 6.5, those three pHs I would consider very important, because these are the pHs where absorption happens in GI tract. And the solubility in those pHs are very important. You might have sulfate salt or hydrochloride salt giving solubility in water 10 milligram, but the minute goes pH 4.5, that depends on pKa. You know the basic fundamentals. That's why you want to influence chemistry or have a chemist to design a functional group which has a pKa greater than 6. This is basic fundamental, greater than 6. So if your pKa is greater than 6, 7, 8, what happens is pH minus 2 plus. So it will be highly ionized plus 2, and below 2 it will be unionized, 50% ionized. So when you have pKa of 6, you'll say, there 50% ionized, unionized, at pH 7, 75, pH 8, highly solubilized, no problem. But if you have pKa of 4, then it's trouble, because after pH 5, it's completely unionized, and you will see drop in dissolution. So Victor, very good point. So solubility of the molecule, in short, pH 4.5, 5.5, 6.5 in physiological conditions, FASU and fed. Those are the important pHs and media to understand the solubility, dissolution, supersaturation, and fraction absorb. Make sense?
Thank you very much.
Thank you.
Any more questions? Okay, I got another one for you. So you talk about locking the solid form early,
yes.
very early, as soon as possible, and that's something that we also advocate. However, there is always a however. Quite often, for instance, the solubility levels that you need for preclinical studies and for tox studies are much greater than what you're going to be needing for clinical trials later on. What are your thoughts about using a form for preclinical studies and then changing it once you go into clinical?
Yeah, no, that's a good point. Means ideally you select the physical form or salt form as early as possible, not the polymorph. Locking the form means hydrochloride salt or sulfate, or let's lock the one salt, because with the small amount of material they could select form and you lock it. Of course, different polymorph could happen. That is the ideal scenario. However, as going back to Victor's point, if we do in situ salt, someone says, what, I don't have money to give you, 30, 40K to, can you just make in situ salt and give me for preclinical? Nothing wrong in that. So what you do is you make a basic compound and add all this acidic counter ion and pick up the counter ion which has very high solubility or dissolution. Of course, look for when it crashes out and how much crashes out, that spring parachute, and pick up in situ salt which has a highest solubility and less crashing with some surfactant and polymer. Once you do that, then you know my molecule is very clean in 2 weeks DRF, in 4 weeks tox. Now you can say, Solitek, can you give me the solid form? Now we are ready to go in the clinic. You could do that also. And there in situ we must have used sulfate salt, but later on we realized hydrochloride is good. No big deal. Just do bridging PK quickly or dissolution, because that won't impact much exposure. Because preclinically you need very high doses. That's why you picked up the highly soluble or dissolved form. And later on, you know, in clinic doses are going to be those. So let's little bridging study and you are good. That's in US, Europe, it's all good. In Japan, they don't want that. Japan, every study has to be with the one form. So just keep that in mind. For Japan, they are a little strict. They said, use your hydrochloride, now you are using sulfate. I need to see with that counter ion those studies. So it's a trouble. So you have to ask, like, are we going in Japan somehow? Yeah, we'll go in 5-10 years, and then we'll worry about that later. You know? But if you are in Europe and US, you are good with that bridging study with the changing the form. But before GLP, I would try to lock, because GLP studies are $1 million, 4-week studies, 9 months readout. You don't want that, you know, affect the dissolution. That's what we do actually, best practice. But it's okay to change later with the bridging PK, and you won't find much difference. Slightly change in dose, you could say. One form gives slightly higher, another gives little low. We'll say, you know, doses could be high in the clinic with this one because we see dissolution, solubility less. That advice you can give. But once you go in the clinic and they see dose linear exposure, you are good. You don't need to worry. Hope that helps.
Thank you very much.
Thank you.
Any more questions in the room?
Hi, I have just one question. What about your experience? Did you see observed transformation of a polymorph into an hydrate during dissolution studies or intrinsic dissolution study? Because it's something that, as you know, can very much affect the solubilization of the drug and the solubility eventually. So I agree with you, you know, better to lock the polymorph, but you know, one thing what you analyze in the solid form, whatever it is, a salt, and then this changes during the dissolution. We have, I'm talking because we had in the past some experience, some diclofenac salts.
Sure.
I mean, I'm sure you have many experience, many examples of this.
No, it's a good point actually. What question I can rephrase, you know, you might pick up the nice stable solid form and suddenly during dissolution solubility it changes to hydrate, it changes to another form. And if you heard me right away, I said select the form which is physically, chemically stable, which means Solitek or any solid state colleague's responsibility to select the form. 99% it will not change. That's the whole point. And if it changes to hydrate, for example, there's some compound, they go into hydrate. Although they form in hydrate, but that hydrate in a solution state is not stable, it goes hydrate, anhydrate, hydrate, anhydrate, and dissolution is still going on, and there is no impact on exposure. You are good. So they will say, well, you know what? This is the best form. It does goes into hydrate, but there is no impact on solubility, dissolution, and exposure. So we are good. So that is important. If that is happening, as soon as you make a nice unhydrate form, and you make it as soon as you start solubility, so trihydrate and goes down, and you'll say, might as well select the hydrate form then. Because anyway you are converting in the body, so that will have impact on dissolution. So you select a hydrate form, and then you manage your technology accordingly to take care of that. So that's smart decisions. That's why solid state is very important. They will tell you what exactly the right one. Hope this helps. Thank you.
Hi, nice talk. I was wondering, like, this thing mainly important if you want to take the drug orally, right?
Yeah.
For example, if you really want to inject, you can use some kind of carrier. So in your industry, like, what's your perspective? Are you slowly moving to use some kind of carrier like liposome or nanoparticle or something like this to deliver small molecule, or it's a big feasibility challenge? For example, I can imagine that there will be storage facility, supply problem. So what's your perspective on that? I think that path will be much easier, no? Like. No, it's a good question. Although I focused on oral dosage form
focusing more on, because 90% of the products are oral for small molecule, and that's how it is. Biologics, you don't need a polymorph, it will never crystallize out, they are amorphous, and there's no solubility problem. So biologics, we are good. This is all NC, and that's why we are talking about oral. However, when you take this molecule into other route, cream, lotion, emulsion, injection, any other route of administration, equally physical form plays a key role. Because there also, if the form is not stable in those media, could crystallize out. It could go into the another form. And that's why early on, when solid state characterization we do and select the form, you won't believe they do like 30, 40, 50 solvents, keep stirring, keep moving. Does it goes to this form, that form, and how long it takes to convert into another form. After doing extensive work, you select the form. And sometime in synthesis, there are some impurities that may lead to another form, which is unpredictable. But 99%, once you select, my experience, I think, in last, of course, in 25 years, in my experience with Novartis, we saw 2 changes like that out of 600 molecules. So there are exceptions. But your point is, when you select a stable form, the goal is it should stay stable anywhere, any dosage form, anywhere. Because that could affect the precipitation, crystallization, stability, and all that. Okay? Thank you.
Thank you very much. Any other questions here in the room? Okay, if not, we're going to thank Sudhakar again for his presentation. We're going to have opportunities to talk to him as well privately when we have the coffee. And thank you very much. And we're going to welcome the next one.
Thank you.